Good morning or good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Kyle from Business Review, and I'll be your host today. It's our pleasure to have Catalent Pharma Solutions with us, who's presenting this webinar entitled Best Practices in Leveraging Phase-Appropriate Expertise and Technologies to Accelerate Your Product Development to Commercialization. Today's guest speakers are Meredith Perry, Director of Pharmaceutics, Catalent San Diego, and Todd Stutzman, Director of Pharmaceutical Development, Manufacturing in Catalent, Kansas City. I'd like to welcome you all to our webinar platform ON24. You'll notice that this webinar is browser-based. So if you disconnect for any reason, please just click on the link you received by email to rejoin the session. If you want to ask any questions, you can send them in via the questions widget. Just type them into the box in the top left-hand corner of your screen, click submit, and we'll have some time at the very end to address any questions or thoughts you may have. Please use the yellow help widget if you require any assistance, and you can move, resize, and maximize any of the windows in front of you to get a better view of the slides. But for now, please allow me to welcome Meredith Perry. Over to you, Meredith.
Thanks, Kyle, and thanks everyone for joining us for the webinar today. For today's webinar, we're going to set the stage by talking about the different goals of early stage and late stage drug development. And then we'll talk about how our early phase and late phase teams have worked together to learn from each other and really use each other's expertise. And finally, we'll share some examples of successful acceleration from early stage to late stage development. So let's start off by talking about what's unique about early phase development and what we can do to be strategic about some of our early experimental planning. So in a lot of ways, early development is really a bad setup for late development, just because the goals of the two types of development can be so different. The goals of early development are often all about speed, so speed to the clinic, minimizing the use of API, minimizing the time, minimizing the cost overall, and really getting drug to patients as quickly as possible to understand the safety and the efficacy of the potential new therapies. So here are a couple of examples we wanted to highlight that kind of show the differences between the goals and the CMC considerations for early phase versus late phase development. When it comes to toxicology data, there often is very limited data early to determine handling practices, and that often means that more conservative handling practices are required early in development, which may limit what types of formulation processes are practical. When it comes to API, often the synthetic process is still very much being defined. It's very common that polymorphs and salt forms are still being investigated, so there may be some uncertainty about what the final form of the API will be. For the dose form, typically a quick-to-clinic dosage form is much more simplified and, again, is all about speed and simplicity and dose flexibility. So powder in a bottle, liquid in a bottle, powder in a capsule, often even prepared on-site at the clinic as an extemporaneous prep, are appropriate for early stage but may not be as scalable. Again, the dose strength range can be quite wide while the safe and efficacious doses are still being identified. So that may limit what types of formulation processes are appropriate. But in early development, you may be able to use a pretty unique product-dedicated or disposable process train to meet the needs of that early process. When it comes to analytical methods, early in development, what you need are just phase-appropriate methods, phase-appropriate levels of qualification or validation. So there's a bit more flexibility. And then when it comes to the knowledge of the whole program, often in early development, a single person holds all of the knowledge. It could be a CMC person at a sponsor company. It could be a CMC consultant. But typically, there's someone who has followed those early stages of development and really knows the history. But throughout the development, the sponsorship of the molecule may change, whether it's a change of personnel at the sponsor company or partnerships with other groups as the compound develops. Management of those knowledge systems will become increasingly important.
As far as looking at the late phase development activities, we're hoping to have more toxicity data around the API to better understand whether engineering controls are going to be required or if, in fact, PPE will be sufficient to protect the operators during the manufacturing process. This is really important because now the batch sizes are much larger. The potential for exposure over longer manufacturing runs increases. So we're really hoping to have this better defined as we go to larger scale manufacturing events. The API ideally is also seeing less and less variability as the process becomes more refined. We also would expect to have very good understanding of the polymorphic forms and controls around those, such that we're further reducing the variability from lot to lot as we scale up the manufacturing process. The dose, at this point in time, we're really hoping to have a very good understanding of the QTPP, or Quality Target Product Profile. Early on, the dose may not be well established, or we may be in a different dosage form altogether. Early phase, there may be a capsule being used based on limited API available at that juncture, but commercially, the targeted product profile may in fact be a tablet to introduce to the market. And at this point in time, there may actually be a transition from one dose form to another. The process will also start to be refined as we look to have a more robust process that can accommodate large-scale manufacture, which would be designed to support the commercial activities at launch. The methods also will need to be fully validated NDA level to support the activities at the later stage. The knowledge transfer, we start to employ some different tools at this point in time where we have a better design space established, and we'll talk about that in subsequent slides. But then also we'll implore risk assessments and further refine the development history.
So let's talk about some strategies that you can use to really get to know your API early in development, while still minimizing the use of API, the quantities, and certainly the costs, which are so critical to early development. One of the first things you can do is to get your compound professionally assessed by a certified toxicologist, and this helps to harmonize the handling across multiple vendors. You may have several different API suppliers, toxicology labs, drug product suppliers, analytical labs, and having well understood toxicology data will help to keep everyone handling the compound safe and may limit the need for you to do repeat assessments across every vendor. So if you can get kind of an independent report, that can be a good setup, and then everyone knows which systems and process trains can be appropriate for the level of containment needed. The next thing to consider is to really try to understand your polymorphs. Even if you can't control your API polymorph early in development, maybe take the time to look for at least the most stable form so that you know you're working with the form that potentially has the most challenging solubility. So really use whatever API, even if it's small quantities, to try to track that polymorph information early. Another thing that's really helpful is to determine where your molecule falls in the DCS classification. And this is even more useful than the BCS classification because the DCS specifically considers the dose strength, which, as we talked about in the previous slide, will change over the life of the molecule. So understanding if your compound behaves like a DCS class 1 based on the dose, or whether the dose strength for safety and efficacy push you more into the 2A or 2B category, those are really important considerations. And that DCS classification can be done strictly as an in silico exercise. So you can just try to predict the permeability and the solubility of the compound using software systems. Certainly getting real lab data is better, but not a bad idea just to collect that data early about where on that DCS classification your molecule falls. So once you actually have some API to work with, it's time to start identifying some of the critical material attributes. And you won't necessarily know what those are, so the early development goal is to collect a range of data to try to make some of those later decisions more well-informed. So when we start on a program, the first thing we ask for is always how much pre-formulation data is available. We're looking for solubility data, we're looking for stability data, and specifically chemical sensitivity to the 5 major pathways of degradation. So we're looking for pH across a typical biological range, depending on the dosage form, oxidative stress, light, heat, and moisture. This is critical to developing meaningful analytical methods, as well as to understanding what some of the processing challenges may be. If a certain process train will expose a molecule to one of these pathways, we want to know about that early. And then the other thing that we'll start to collect is the physical characterization data. So the tests listed here, PXRD, TGA, DSC, SEM, and GVS, are really material-sparing ways to get a lot of good information. So all 5 of these tests can probably be done with well under 100 milligrams of API. So I really think it's a best practice to characterize every lot of API prior to its use. And this is true even for preclinical lots, because the solubility and the absorption variation are really important to be able to understand. So even if you're just making a gram of material at lab scale and using it in some very early non-GLP PK studies, see if you have enough material to collect a PXRD spectrum. Understand if it's the same crystal form from lot to lot. If you get just amazing solubility or amazing absorption from one particular lot, and it turns out that lot was the only lot that was amorphous compared to other lots being crystalline, that can be really important data in building the history for that particular API.
Yeah, and just to further expand on Meredith's points there, you know, the understanding of the polymorphic forms are really going to become critical as we increase in scale and potentially modify some of the process. So the understanding is now becoming critical such that we don't cause a polymorphic shift to occur and ensure that we maintain that same crystal form. Particle size, you know, that's one that typically we see changes in as a function of scale in API and as the synthetic process starts to evolve. This is really very important to multiple aspects of the finished dosage form. It can impact the content uniformity, the dissolution, flow characteristics, all very important. Same with surface area. Also has a relationship very much like particle size, in which it can impact dissolution. If in fact that compound is hygroscopic, the rate at which it picks up moisture. It can also impact the lubrication efficiency of the granulation. All of these things are very, very important for a successful scale-up event. And so, you know, even things as simple as bulk density are important to have characterized from lot to lot so that we're aware of any subtle changes in the API and can prepare for what differences may result from that particular change as we manufacture larger batches. The wettability, also, you know, an aspect that's extremely important. We've seen this time and time again, actually. There's a program that we're currently working on in which the drug is highly soluble. In fact, it's very hygroscopic and deliquesce at room temperature if left out for 6 hours. But that particular tablet has disintegration times of 16 minutes. This is largely in part due to the wettability of the API, and by the addition of a surfactant, we're able to take in and address those types of issues. Bioavailability, understanding whether or not there's a high first pass effect with the absorption of the API, and how much is actually making it into the bloodstream, can really impact how we take and administer the finished dosage form. Maybe a buccal tablet would be the preferred administration in order to bypass the level of metabolism that's seen through oral consumption. So all these things need to be taken into consideration as you're continuing to develop the finished dosage form.
Now let's talk about some strategies to approaching formulation development that, again, bridge that gap between early development goals and the long-term, larger scale product. So as we talked about, we really want to use all of that API data to drive our decisions. The DCS classification, the pre-formulation data, and the physical characterization will all guide us toward what type of dosage form would be appropriate, both in the short term and the long term for the compound. The other thing that's important is knowing how reproducible your API is, because that can help to determine how sophisticated the formulation needs to be. If the API is very consistent, it's always the same crystal form, or if it's always amorphous and that's consistent, that can help us to make our formulation decisions as well. One thing we'd like to recommend is that excipient compatibility is really important. Even if it's not on the critical path for early development, it's still worth performing a thorough excipient compatibility study because it will support the scale-up work down the line. There's not always a lot of time in between early development and the need to scale up quickly for compounds that are showing good efficacy. And so at that point, you don't want the excipient compatibility to be the rate-limiting step to starting that scale-up work. So we would recommend screening at least 2 excipients from each functional class, so fillers, binders, disintegrants, lubricants, etc. That way you have a couple options if one particular excipient shows a challenge. You want to pick relevant excipient ratios, and this certainly can be challenging because you don't yet know your final dosage form, your final dosage strength, but put some thought into what an appropriate excipient ratio would be and what a relevant storage condition would be for this product, knowing that you have some early API stability data. Think about what a long-term storage condition could look like. And also consider how you're actually creating the samples for excipient compatibility. Is a blend appropriate? Would it be better to make small compacts? If you're using API, make sure its crystal form is appropriate. If you're using an enhanced intermediate like a spray-dried dispersion, use the spray-dried dispersion for the excipient compatibility because it will likely be more reactive than the crystalline API. So definitely collect that data, even if you're doing it in parallel with other development. And then consider what your downstream process train is going to look like so that you can start to make formulation decisions that would support the long-term product. One example would be adding some tablet excipients to a capsule formulation. If you know that you eventually will have a compressed dosage form, even an automated capsule, you will need a lubricant. So maybe include some lubricant in your early capsule formulation. Even though it's not a critical processing aid at that scale, you will already have some short-term and potentially long-term stability data with a mixture of excipients that contain all of the critical functional classes. Another example would be to use an instrumented tablet press or compaction simulation in early development to understand the robustness of your formulation. On small scale, it's possible to make almost everything work just once, but the repeatability is just not often well understood at small scale. And it often can't be because there isn't enough API to repeat processes multiple times. So by using instrumented data, you can at least collect that background data and start to understand what some of the formulation challenges may be. Is the formulation sensitive to speed, to pressure, to ejection force? Those are the kinds of things you can start to understand even on small batches. Another example would be picking a small-scale process that really doesn't scale well. So if you're using something like sonication to dissolve a critical excipient or API in a critical fluid, that's probably not a process that will scale. So not to say that you can't use sonication at a small scale, but make sure that you keep in mind what will and won't scale and start to put in some thought to what process trains would work later on. So a big part of being able to conduct a successful development program is documenting the decisions along the way. As we mentioned earlier, often in early development, there's only one, maybe two main points of contact, and the information is just known by the people who have worked on the project all the way through. So documenting the development history is not so much about the formal reports, but it's about how easy it is to transfer data to new parties. This could be internally within a sponsor company as personnel change or as the team grows. It could be externally to vendors. It's not about how formally you document things, but how easy it is to share that information and to share the why, not just what was done, not just raw data, but what the decisions were. Maybe the dose strength changed as a result of some early toxicology data, or maybe the formulation pivoted because you learned something new about your patient population. Maybe you knew about a risk and you took some action. So document all of those things so that it's easy to find, clear what was done in what chronological order, and that will make the program more valuable even to future partners because they will be able to understand what was done and potentially what's missing. So at this point, I also wanted to mention that all of this really fits the quality by design mindset. It's about being systematic. It's about defining your objectives, understanding that the objectives are different in early stage versus late stage, but making all decisions based on sound science. So starting with the API and following all the way through to the formulation, we really want to make data-driven decisions the whole way. So when it comes to strategies for analytical methods, again, there is a lot of difference between the goals of early development and early analytical methods and later stage analytical methods. So you want to use those critical material attributes that you've started to collect on your API to accelerate the method development. As we mentioned earlier, understanding those 5 pathways of degradation is really critical. It's the foundation for everything else in your analytical programs. Don't skip the forced degradation work, and especially look at both the solid state and the solution state for both light and heat. I think often in analytical method development, you're mostly thinking about analytical samples, but by collecting data on both the solution state and the solid state, you can keep in mind possible process exposures as well. When it comes to the qualification level for analytical methods, early development is kind of unique because you can be a bit more creative and really use discriminating methods to distinguish among formulations. The methods don't really need to be standard pharmacopoeial methods. You can use creative equipment. You can use systems that would not be validatable because that's not the goal of some of the early analytical methods. You're really looking to understand the API and distinguish among formulation platforms early. When you get to the point of starting to qualify the methods, focus on the science. Really use the critical attributes: specificity, sensitivity, accuracy, linearity, precision. Document the results. It's not so much about setting specifications. It's more important to understand what is the expected performance of the method. How repeatable is it just in day-to-day use to start to build that knowledge base? A qualification may be very phase appropriate as opposed to a validation. And this understands that the late phase site will need to optimize the method anyway. The formulation will change, the type of product may change, capsule to tablet or solution to tablet. The dose strength will certainly change. So it doesn't make a lot of sense to commit a lot of resources to validating methods very early in the development process, knowing that it will have to be repeated later. But again, use good science to make those decisions about where to focus the time and the budget for the program. You do want to consider that the method needs to be appropriately robust. And what I mean by that is that you may have multiple vendors using these analytical methods. So some of them may have HPLCs and some may have UPLCs. Some may use one specific manufacturer of their chromatography systems. So the method needs to be appropriately robust to work across multiple vendors. I'm not talking about a formal robustness evaluation, but keep in mind that it does need to be transferable. And so with a well-qualified, scientifically sound method, you can get good stability data. It doesn't need QA review at most stages. You can keep it pretty simple, keep the cost a bit lower early in the program, but still collect really high-quality data. And at the end of all this, just make sure that you document any sort of gap analysis. So if there's something you have not done or is not appropriate to do at early stage, make sure that's written down so that whoever takes on the program later knows what the gaps are and where effort will need to be focused. And then when there's time during clinical development, you can address some of those gaps. So impurity qualification is one of those examples. It's not expected that every impurity will be well known and well qualified early in development, but start to track the history of the impurities lot to lot and any related substances generated on stability so that it's easier to go back and understand that data later if it's very well organized and well documented.
So next we're going to talk a little bit about some of the collaboration efforts from site to site and how we go over the process development, working with the San Diego site, and how we've harmonized a lot of the capabilities, and then finally we'll end with the transfer of information and knowledge. So the first thing that we need to get a very good understanding of is the material handling. The occupational exposure limits, and Catalent has harmonized across all sites to a 4-band system, with 4 being the most potent, and that is less than 1 microgram per cubic meter occupational exposure limit. And there are certain sites that actually can accommodate compounds in that particular area where other sites are not capable and only go to a band 3. The other thing that we really need to have a very good understanding of is the onboarding and the proper storage of the raw materials. The documentation that is required to take the program to the next level and be manufactured in a facility which has 80 countries worldwide that we ship to, and the regulatory expectations associated with that is maybe quite different than when you start with phase 1, phase 2 activities. So we need to make sure that that documentation is up to the standards that are required for that particular site. We'll also want to take and get a very good handle on the environmental controls, humidity, light, temperature, sensitivity, those things that Meredith was discussing previously, and whether or not we need to take and implore additional engineering controls. We need to have a CapEx and acquire equipment around that to ensure that we are able to maintain the same quality of product as we go up in scale. The pre-formulation characterization, that information is critical. And the San Diego site does a very good job of assimilating that information and then transferring it to us, and we'll cover that in more detail in one of the subsequent slides. And then the last are the process considerations. So, the San Diego site has predominantly, at this point in time, V blenders, and in the Kansas City site, for diffusional blending, we use Ben blenders for the most part. We do have V blenders as well, but this is one of the transitions that typically will occur, and the reason for it is kind of back to that occupational exposure limit. The transfer of materials in a closed system are really preferred by the regulatory bodies. It eliminates the potential introduction of foreign materials, and it also provides an additional level of safety for the operators. So understanding the development history. We really do take a lot of effort to talk with the scientists directly. The early phase scientists have a great amount of information that is crucial to the successful transfer to the other site. So we do spend a lot of time discussing with them and getting a very good understanding of the critical material attributes, the pre-formulation data, the physical characterization, all of those things which are going to make the transition successful. The CPPs, there may be some initial thoughts on what a CPP might be based on the scale that they're currently manufacturing. So we'll look to them to get indications as to what to look for and what to capture additional information around. And then really it's data that's going to drive the decisions on how the program moves forward. So API is almost always at a premium, and there's a limited amount for development non-clinical activities. So one of the things that we try to do is minimize that consumption at the tech transfer event. We do like to keep that batch to be representative of a clinical batch. So we like it to be no less than 1/4 the targeted clinical batch. This further reduces the risk associated with the manufacture of that clinical batch because we quite often will do multiple subs at the same scale to accommodate that clinical batch. Usually we're operating in the 1.5 to 3 kilo range for this tech transfer. That's kind of the lower end for what we like to do. If there's more material available, we would do it larger, but this is the bare minimum typically. And this is a typical equipment train that would be used for this event. A 10-liter bin blender, the Gerteis mini pactor, the Korsch XL100, and then coating in a perforated CompuLab 15-inch pan or 12-inch pan, depending on the batch size. For this batch, we're going to take and we're going to collect BUs using our micro sampling units, and then in addition to that, we'll pull some stratified CU from the beginning, middle, and end of run to further assess any potential segregation as a result of from full hopper to emptying the hopper over that compression run. We'll also take and look at the potential for segregation as a result of fluidization as we transfer that from the bin blender in a closed system and dropping that material into the hopper of the Korsch XL100. Just as important as the equipment is having a harmonized capability around in-process testing and finished product testing. And so we've worked with the San Diego site to ensure that we're aligned from the types of tests that are done, but then in addition to that, also the instrumentation that's being used. So, quite important, flow analysis. San Diego uses the Freeman FT4, and we have a Freeman FT4. So the database that's established early on with San Diego can continue on in Kansas City as we start to scale batches and look to process refinement and improvements. In addition to that, AccuPick, GeoPick to test for true density and envelope density and establish solid fractions, same instrumentation at both sites. As far as characterizing, for example, just core tablets as an example here, the standard battery of tests are done at both sites in the same manner.
Yeah, and
in addition to
one more point is just, sorry, Todd, one more point is just that I think it really helps that we have harmonized not only the equipment we have now, but also what we want to upgrade in the future. So as we have talked about our current capabilities or as new technologies come online, we talk about our wish lists and figure out how we can harmonize or work together to build those databases and really improve the testing capabilities and certainly the formulation equipment going forward as well.
Yep, agreed. So for common core equipment at a small scale, we do have comparable equipment, or if not exactly like for like. The high shear granulators used in Kansas City are all vertical bottom impeller, horizontal chopper. They have the same configuration in San Diego. San Diego has Gerteis Mini Pactor. We have Gerteis Mini Pactor. The Korsch XL100 for initial tablet development, excellent rotary tablet press, fully instrumented, that will give us some of that meaningful data as we start to look at lubrication efficiency, compression profiles, things like that, and then dwell time sensitivity will be on the same type of press that was employed in the San Diego site. For encapsulation, dosating disc, Bosch-based units, we have those at both sites. And then for coating, standard perforated pan type of coating platform at both sites. So we really hope to kind of narrow down what we think are CPPs based on the information that we get from San Diego. And several of our scientists are trained in STADI software. That's the software that we use to do our fractional factorial design, which is the tool that we commonly use in the design of the process space, and to better understand that. So those batches are typically done at 2 to 5 kilo scale, depending on the process train. And this is really important for us to take and be able to confidently provide a filing that we can stand behind and feel like the design space is well established and understood, and then also provide the basis by which we scale up from. The analytical testing that's done at this level is limited typically to, we're looking at dissolution. We may have some blend uniformity depending on the batch size and drug load, but we will typically look at content uniformity from beginning, middle, and end of run, dissolution, and moisture analysis. And then this would be an example of what a study might consist of in further establishing those CPPs. In this particular case, we have a process flow here of a standard roller compaction program. And to the left, you are looking at some factors, pre-roller compaction blend time with lubricant. And why that would be important. The lubrication efficiency that you achieve there can then translate into how those ribbons fracture during the milling step and translate into different particle size generations. And so that's, a factor that we like to incorporate in the overall design, because then that translates down into weight uniformity as a function of press speed. Also can impact, how compressible the tablets are and the tablet hardness that will result from maintaining a constant force being applied during this study. We'll also want to look at things like pressure, the roll gap, and even roll speed. Roll speed, we're going to look at that just to ensure that we're maximizing the throughput and having the most efficient process. Roll pressure, roll and gap setting, they're going to help us confirm, what is the appropriate solid fraction. Do we have the right porosity level? And in fact, is the formulation sensitive to that porosity level from a lubrication efficiency standpoint? So all of these things will be monitored and evaluated in this particular study. And then the responses are responses that you would typically see, such as flow rate. We'll look at bulk tap density. We're going to look at the sieve analysis of the resulting granules. We're going to look at weight uniformity and the standard deviation of the tablets that are produced. Content uniformity, dissolution testing, and is there any of the factors that are impacting that? Tablet hardness, the friability, disintegration time, the acceptable yield, and moisture, if appropriate. And this is just an example of a roller compaction tablet process. So for the knowledge transfer, I think this is really important, and this is from site to site. In a little bit, we'll also talk about from site to client. But in this particular case, we're fortunate enough that we have a program manager that has spent time at both the San Diego and the Kansas City site and does an excellent job of facilitating the transfer of information. I mean, we start with the upload of all these documents from, the safety and cleaning verification and API data, development history, process development, methods, batch records. All that gets uploaded to a team folder that the scientists at both sites have access to. In addition to that, we have several teleconferences, and we even have the scientists moving from site to site for manufacturing events to see the activity firsthand. This is very, very important because, as the saying goes, a picture's worth 1,000 words, and the experience that you get firsthand seeing it is extremely valuable. On the
Absolutely.
resulting side,
I think also, sorry, Todd, go ahead.
No, go ahead.
So the other thing that I think has been really valuable is the informal collaborations. Just getting on the phone and having scientists talk to each other, or having a person in plant to observe processes during that transfer. As you said, there's no substitute for seeing it, and even for kind of seeing the body language. So we have learned there are certain excipients that we use in early development that really make a late phase development person squirm. And so we've learned a lot about, even if there are published data about a particular excipient being suitable, there may be some unique processing challenges that the operators who work on large scale can really translate. And I don't think we would have gotten that if we hadn't worked so closely together.
We really appreciate that, Meredith. And yeah, I think, we're definitely making a lot of progress with successful transfers from site to site. We've seen a lot of progress. The other thing is the teams and the way the teams are matching up and transferring that information from project manager to project manager, the pharmaceutics team, but then in addition to that, the analytical team, EH&S participates in this, procurement, QC and QA both in making sure that the appropriate documentation is being provided to facilitate a smooth transition and an efficient transition. So once we've made the transition to the Kansas City site, the other thing that we start to put together are a format of information that we like to share with the client on a periodic basis. One of those formats is what we refer to as the moat, a term actually used by a former client, which refers to the mother of all tables. It stuck and we continue to use it today. But if you look at this is just an example of, say, a roller compaction process that is being done over multiple vendors of API, which is commonly seen with programs as the scale increases and the requirements for API increase. And what we're looking at in just this tab is how those various lots of API impacted the ribbon true density, envelope density, and then resulting solid fraction over time. And it's a database repository of information. Each tab has a different stage of the process in which we collect all this data and can look at it from the beginning of the program over the life of the program. Another example here is one just for the API, and there's always a tab for the API in which we're going to look at, the standard battery of tests that we've talked about previously, the bulk tap density. we're going to look at the flow analysis. We'll look at sieve analysis, and then if there's Malvern data or laser diffraction particle size data, we'll also incorporate that. And once again, building that database by which we look from lot to lot variability and get ahead of, if in fact there are changes that can potentially impact the process formulation, we're ahead of the curve and able to raise the warning flag and say, wait a second, we're seeing some differences here that we have concerns about. And make sure that we put the proper mitigation plans in place to address it. Another tool that we like to use is the quality risk assessment. And so this is where we take and look at the whole process from beginning to end, API included, and we take and break it down into the probability of the risk occurring, the detectability, and then the severity. And each one is assigned a value depending on the stage of the risk. What we try to do is then share this with our clients and make sure that we are aligned on the risks that we see ahead of us, and then we work with them to put in the proper mitigation and contingency plans to address this. Now, this is a live document that changes over time. So as we complete additional development activities and have a better understanding of the process and formulation, for example, process optimization for DOE, some of the risks go down and are reduced significantly. And that's where we try to take the program as it evolves. A unique offering that we have in both San Diego and in Kansas City is what we refer to as Project Glass. And in the current environment where travel is limited or prohibited altogether, this is a great tool for not only the scientists at both sites, but also for our clients. This gives them the firsthand view of what the scientist is seeing. And so they wear this headset, which streams through a secure app back to a laptop of either our fellow scientists or our clients, and they can see and audio communicate with the scientists firsthand. And we found that, especially in today's world, the popularity of this has gone up dramatically. And so now we're going to transition to some lessons learned. We have some supply chain management issue, a couple case studies, and then we'll talk about the integrated solutions. And so one of the things that are really important are the alignment of the raw materials. And this really is key in accelerating programs and ensuring that we don't hit a speed bump or a misstep as a program takes and transitions from one site to another and goes up in scale. So we want to ensure that we're using, the same grade, particle size, vendor, the same site of manufacturer, and that those sites are qualified. There are some sites, where there's a single source excipient, and we need to be aware of that and make provisions ahead of time to ensure that both sites have that vendor qualified. We recently ran into a situation in which Avicel was needed for a registration batch that was a unique grade of Avicel. The vendor was qualified at both sites, but the one site did not have any in stock. And therefore, in order to take and meet that very aggressive timeline and get the registration batches online, we were fortunate enough that our San Diego site had some and were able to provide it such that we were able to manufacture that registration batch on time and get it on stability when the client needed it.
So to talk about one specific example There was a client that came to the San Diego site with what they said was an extremely hygroscopic API. We transferred in a process train that included a simple dry blending, dry granulation tablet, so pretty straightforward process train, but we needed to maintain a very low level of relative humidity. And so on a small scale, we were able to use kind of local soft wall containment, so flexible containment, and then purge that space with an inert gas to control the humidity to keep it well below the threshold that would be problematic. And while this worked at a relatively small scale, it wasn't going to scale very well to larger equipment, and the consumption of the inert gas could have been problematic at a larger scale. So this is where we sort of handed off the program to the Kansas City team and worked with them to design a more scalable solution.
Yeah, this is one of my favorite case studies, actually, and so we actually sent a scientist to San Diego to see the process firsthand and get a very good understanding of how sensitive this API really was, and it is extremely sensitive to environmental moisture. In addition to that, we consulted with the San Diego site on, what we were thinking might be a solution and got their buy-in as well. And so a very proactive approach. We started discussions several months in advance of the program actually transitioning to the Kansas City site. And at that point in time, we were able to make provisions. So we're fortunate the Kansas City site actually has an engineering group and a fabrication group on site. And they were actually capable of taking and incorporating a unique system that provided environmental local controls to the various unit operations through the administer of compressed dried air. This allowed to manufacture without the flexible containment, but still maintain the same level of humidity control required in a localized manner. And so that particular program has gone successfully and is continuing on.
So the other example we wanted to mention was a case where the dose strength range was very wide, moving from early development to later development. So the San Diego team worked on a program where the dose strength range initially was 1 to 8x the target, and this was a tablet product. But for later phase work, the client needed a 40x dose strength. So again, the range of dose strengths was pretty challenging. This needed to be a pretty high drug load product in order to get to those very high dose strengths, and it was one of the most compressible APIs our scientists had ever seen. So we tried a couple different powder feeding designs on the tablet press. We tried to collect some background rheometry data and really tried to document what some of the challenges were at small scale so that anything we learned on the lower dose strengths would help the larger scale site, the Kansas City team, to be able to make those higher dose strengths without some of the challenges we were starting to get hints of. Powder flow challenges, as I mentioned, compressibility, and starting to see some picking or uneven surfaces on the tablets. And so by identifying some of those challenges early, we were able to then make a better handoff to the Kansas City team.
Yeah, so this particular program, we were very fortunate because those insights that the San Diego team provided allowed us to take and come up with a larger scale solution that we employed. And in this particular case, we took the highly compressible nature of the low density API and used it to our advantage by going through multiple roller compaction events to reduce the overall surface area of the API, but also densify and improve the flow of it, coupled with the fact that it was highly brittle fracture. So we weren't as concerned about going through those multiple roller compaction events to then translate to compressing on a high-speed rotary tablet press. Another program that we we're able to transition smoothly, meet the client's needs of the higher drug loading and higher dose tablet formation.
So as we've talked about throughout the presentation, the San Diego site really focuses on early development. We've been doing early development for more than 20 years now and really focus in that preclinical through phase 2 space. We can do all of the analytical, the development, the manufacturing, the packaging, and labeling at one facility, but really focusing on being flexible and being able to adapt to quick timelines, changing dose strength, all of the things that come with early clinical trials. We previously were known as Pharmatech and joined the Catalent team about 3.5 years ago. And so I think this really has allowed us to leverage what we have a lot of experience in and acknowledge that we would probably not have been able to do both early development and late stage development to the level of excellence that we require of ourselves. And so I think it has been a great collaboration.
The Kansas City site has a pretty rich history of being a big pharma site. Originally started as Marion Labs back in the mid-'60s and went through various mergers, acquisitions, and such throughout that time period. In 2000, it transitioned to a contract manufacturing development site. And so the site has it's an integrated site. We have the analytical capabilities, the formulation process, and clinical manufacturing, plus commercial manufacturing, and then packaging all on the same campus. To date, we have 13 commercial products that were developed in Kansas City that are now on the market. There are 2 additional that have completed validation and are awaiting approval, so soon to be 15. We ship to over 80 countries worldwide, and we have a very, very pristine inspection history. So I think there's a high level of confidence and comfort in the quality systems that reside in Kansas City, and knowing that your program will not be put at risk from a quality aspect.
So in the 3.5 years that Pharmatech has been now with the Catalent team and now the Catalent San Diego site, I think we've really built some great efficiencies together with the Kansas City team to make the transitions between sites as seamless as possible. It's not just the technical expertise, although that has been a great benefit. It's the people working with people to really get to know the ins and outs of what the different goals are for the different sites, and that really has helped to make the technical transfers go more effectively. As Todd mentioned earlier, we also have a dedicated program manager for any projects that transfer between sites, and being really proactive about assigning project managers at both sites helps us to integrate the knowledge before we even begin a transfer. We can do a lot of activities in parallel, which creates a lot of efficiency. Things like safety assessments are already harmonized. Things like cleaning methods can be started in advance. We harmonize quality technical agreements as much as appropriate across the 2 sites. We can work on ID methods, API shipping, and so on. And so that really helps to save time in those critical transitions between clinical phases. And overall, the goal is to avoid any sort of rework to spare API. We know that API is precious. We know that time is precious. We both have the same goal of getting product to patients to help patients as quickly as possible and with the highest quality possible. And so all of these things really help us to achieve that goal. So I hope that throughout this webinar, we have showed the value of really using that late stage development experience to inform even our very early drug development. If you work with multiple vendors across the journey from preclinical all the way to commercialization, there can be a lot of handoffs in the project from site to site or from vendor to vendor if you have multiple vendors. And that creates risk in the program. Development of drugs is risky enough without any additional risk added to the program. So by working with one partner like Catalent all the way through the process, you can certainly reduce the risk and enhance the likelihood of success of the molecule. We can work quickly. We really have worked hard to accelerate timelines by doing proactive work, by doing work in parallel, and by transferring knowledge. We can reduce the risk in a program by, again, aligning our documentation systems, our personnel, our technical capabilities. And overall, we can just simplify your experience of drug development. It can be something as simple as having one conference call with 2 different teams, a single project manager, a single point of contact. I think in this day and age, a lot of us get conference call fatigue and just trying to figure out everyone's schedules and make sure all the knowledge is transferred. When you work with a single vendor, it's really easy to just get everyone on the phone. That way, the early stage team knows the late stage team's goals. The late stage team hears some of the critical decision making early, and you don't have to repeat yourself. So it just simplifies your overall vendor management, which helps you to, as a sponsor, to be as conservative as possible with your overall budget. So thank you for joining us today. Thank you, Todd, and I think we'll turn it over to questions.
Thanks, Meredith.
Okay, thank you both. It is now time for the Q&A. So a quick reminder on how this works. Just type your questions into the box in the top left-hand corner of your screen, click submit, and then wait to hear if your question's read out. But we've had quite a few come in, so we're just going to get started with what types of in silico tools are useful in drug development?
That's a good question. I can start with a couple examples. I think there's certainly a lot of discussion about what type of software systems may be helpful, again, to try to spare API and save time. So the first one that we mentioned earlier was about understanding your molecule. So just the molecule prediction software to understand the log P, the expected solubility. That's a great place to start. You can use that to plug into method development software, so knowing the characteristics, the chemical characteristics of the molecule can help you to potentially accelerate method development. So rather than doing everything by trial and error, using method development software, and there are a variety of different options available, can help to accelerate some of that early preclinical, pre-formulation and method development work. And then we've also had really good luck with doing the PBPK modeling. Catalent has used GastroPlus to do PBPK modeling, and that really helps to bridge that early preclinical dosing with the in vitro results that we're collecting. It's not exactly an IV-IVC, and that's not the intent. The point is to make well-informed decisions. And if you can use the software to help determine what's the critical parameter for absorption, is it particle size, is it solubility, is it permeability, is there an efflux mechanism that you really are not going to be able to overcome or saturate with formulation, you want to know that early before you put a lot of effort and a lot of API into developing a very sophisticated formulation when there may be a different approach. So those are some of the initial in silico tools that I can think of.
Okay, thank you very much. I've got another question here asking, how can small startups prepare to play in the right CMC activities or workflow?
That's a good question also. I think we talked a lot about API sparing. Certainly ask your vendor what techniques they have, whether it's smaller scale equipment, in order to minimize API use and minimize time and cost for early development, and really document all of those decisions. So if you're using very small equipment, that rationale and that history, the development being written down is really critical. Todd, anything you want to add?
No.
Okay, very good. I'm afraid that's all the time we're going to have for today, which just leaves a big thanks to Meredith and Todd for the great presentation, and of course to Catalent Pharma Solutions for sponsoring this session. We did get a lot of good questions, so if it wasn't read out today, you may be emailed later on, so keep an eye out there for a more in-depth answer. And again, to everyone watching, you will be receiving an email shortly letting you know how to access the on-demand version of this webinar. Or you can always watch it through our website, which is www.business-review-webinars.com, We look forward to sharing further webinars with you, so please do keep an eye on the website just mentioned. And follow us on Twitter at DR Webinars Daily Updates, and join our LinkedIn group, Business Review Webinars. Thank you all once again, and I hope everyone enjoys the rest of their day.